Thank you so much for everyone who's tuned in today to hear me tell you a little bit about how we've been using top-down proteomics for deciphering hypertrophic cardiomyopathy. So I'll start off with an introduction to top-down proteomics, since I'm not sure how familiar everyone is with mass spectrometry-based proteomics, but the specific type of proteomics we do is called top-down proteomics. And then I'll talk to you about how we've been using this technique to study hypertrophic cardiomyopathy, which is a really common heritable heart disease. And then I'll finish up by telling you a little bit about the technology development that I've been working on over the last couple of years for characterizing large proteins using top-down proteomics. And then I'll finish up with some conclusions and future directions for how we're moving from analyzing tissue from these hypertrophic cardiomyopathy patients to hopefully modeling the disease in cell-based platforms. So just to tell you a little bit about what our group does and giving an overview, we use high-resolution mass spectrometry-based proteomics and metabolomics, and we work at the interfaces of chemistry, biology, and medicine to study topics such as muscle biology, cardiac signaling, heart failure, and regenerative medicine. Specifically, I'm a chemistry PhD student, so I've worked a lot on method development for top-down proteomics, and I've used that recently in the last few years to study hypertrophic cardiomyopathy and kind of bridge that interface. So it's been a really nice, unique experience to be able to use the tools you develop to study different diseases. So telling you a little bit about what top-down proteomics is, it provides a bird's eye view of proteoforms in biological systems. So proteoforms come from, they're the closest to biological processes, and so we really care about studying biological systems at the level of proteoforms. In addition, they diversify the functionality of our proteome. So even just from one gene, we can get a variety of different protein forms or proteoforms. And that comes from different processes such as genetic mutations at the gene level. We have at the transcript level, you can get alternative splicing or RNA editing. And then after translation, you can also have various post-translational modifications that diversify the functions of protein products from one single gene. So I'll show you one of my favorite real data examples here. And if you're not familiar with reading a mass spectrum, I'll just tell you all that you need to know right now is if you look at the x-axis, you can see here it's just a mass axis, and each peak here represents a proteoform. So that's all you need to know to understand the first half of this talk. And so here you can see that there are proteoforms that come from wild type and mutant from a single nucleotide polymorphism. So you can see an example of a proteoform that comes from the gene level. And then you also see that there are two different alternative splicing isoforms that arise from this gene, cipher 6 and cipher 5. And then also you can see their associated phosphorylated proteoforms. So all from one gene, LDB3, we can see this complex 8 different proteoforms. And not only that, we can see these proteoforms in our samples, we can measure their abundance and compare them between healthy samples and disease samples and start to understand how these proteoforms change in health and disease. And then by top-down proteomics, what we can do is we can isolate any given proteoform. We can use tandem mass spectrometry to get sequence information about it, so we can sequence the primary amino acid backbone. And we can also use that information to then localize post-translational modifications or any sequence variant that might arise from a mutation or from alternative splicing. So that's a little overview of top-down proteomics, but I really want to drive this point home for how we can use high-resolution mass spectrometry to really unravel the complexity of our proteome at the proteoform level. So if you look here, I'm showing you just an SDS page gel of a purified cardiac troponin I protein. So just looking at the gel, you can kind of compare this to if you were looking at the top of an iceberg. So what you can see above the water would be this band in this gel. So you can see the cardiac troponin I here. But if you were to infuse this into your mass spectrometer, you actually see that there's much more there than just one single protein. There's actually over 30 different proteoforms that arise just from this protein that it seems like there's just one, but actually we can really start to unravel the complexity that we would see here just from one band in a gel. So this gives you kind of an example of how we can use this technology to really dive into our biological systems. So we were really interested in studying a specific disease called hypertrophic cardiomyopathy, which is commonly referred to as the disease of the sarcomere. It's one of the most common heritable heart diseases and a cause of sudden death in young adults. And it's been linked to mutations in genes which encode sarcomeric proteins. And over 1,400 mutations have been identified in at least 11 different sarcomeric protein genes. So here I'm just showing you a cartoon schematic of the cardiac sarcomere. We know that proteins interact with each other and they signal through phosphorylation events to make our heart contract and relax. And so even though and also I'll show you that I'm showing just a few genes, the frequency of the mutations in just a few sarcomere encoding protein genes. And so we wanted to know, even though the genetic basis of this disease has been well studied and is pretty well understood to be linked to a lot of different sarcomeric protein genes, how can these mutations in different sarcomeric protein genes result in highly similar phenotypes? And then additionally, we wanted to ask the question, what is the relationship between genotype and proteoform profiles? So we really wanted to measure these different sarcomere proteoform profiles in heart tissue to understand how they change and what the relationship is to different HCM genotypes. So we have a collaborative international team. So starting from UW-Madison, we have this team led by Inga, who's my PhD advisor, and we have a variety of different cardiologists and MD PhDs working with us on this team here. And then we also collaborate with Dr. Steve Marston and Peter Vikarev from the National Heart and Lung Institute in London. And also we work with the Sydney Heart Bank in Sydney and Dr. Chris Dos Remedios and Sean Lau and Amy Lee. So we have this nice international team that we've worked together to collect tissue that we can analyze using our top-down proteomics methods to sort of start to answer those questions that I was posing in the previous slide. So for the HCM patient tissue samples, we were able to obtain 16 different samples from HCM patients. So if you're unfamiliar with HCM and the treatment for HCM, there's actually, this disease is characterized by a thickening of the left ventricle wall and also the septal wall. And so patients who come in, they can undergo this treatment called a septal myectomy. And this tissue is what we've actually obtained for analysis. So we have obtained these septal myectomy tissues, and you can see it's from a variety of different genders and ages. And we actually have information about the mutations for at least 8 of these samples. And then we were also able to get our hands on some donor heart tissue. So donor hearts that were unable to be used in transplant, we obtained those, and we were able to use those tissues as a comparison to the HCM tissues sort of as our control, so we can see how these proteoforms are changing between diseased tissue and non-diseased tissue from donors. So just to go over our workflow, what we did was we cut 10 to 15 milligrams of tissue. We homogenized those tissues, and we were able to extract the sarcomeric proteins specifically. So we obtained an enriched fraction of the sarcomeric proteins for analysis. I normalized those samples to 100 nanograms per microliter so that we can keep everything quantitative. So we want to compare apples to apples here. And we analyzed those using LC-MS. So the type of liquid chromatography that we used was reverse phase chromatography. So we separated all the proteins in our mixture by their hydrophobicity and then analyzed them using a quadrupole time of flight or QTOF mass spectrometer. And this was a really robust liquid chromatography platform, and it was very highly reproducible, which we really care about if we're trying to do any type of quantitative comparison. And it also took less than 3 hours per sample. So I'm showing here again the cartoon of the cardiac sarcomere just so I can show you what we actually could see after this analysis. And I worked really closely with Dr. Wen-Chuan Kai, who's a former group member. She graduated 2 years ago. And you can see here that in control and HCM samples, I'm just showing you a representative reverse phase chromatogram. So you can see each of these peaks represents MS signal. So you can see that we actually were able to detect a lot of these sarcomeric proteins that you see here in the schematic. And then looking closer at the mass spec part of things, so just to remind you, when you're looking at these type of mass spectrum, you can see that each peak would represent a proteoform, and you can see that the x-axis is the mass, so you can just measure the mass of each proteoform here. So just to tell you a little bit about what we're seeing here, you can see cardiac troponin I, which is CTNT, and its phosphorylated forms. You can also see truncated forms of this protein. You can see cardiac troponin I, tropomyosin, myosin's light chains 1 and 2. And you can also see a lot of different zetas proteins, which is a cardiac signaling hotspot. So we really care about seeing these proteins and their phosphorylated proteoforms as well. So you can see that I'm highlighting a few of the limb domain proteins actually, because we were able to actually, these were very well represented among the proteins that we were able to detect with this method. And not shown here, but PDLIM3 and CRIP2 were also among the proteins we were able to detect and measure. So now that we're able to detect these proteoforms, we can also measure their abundance relative to each other between different samples. And so I just want to go into this just a little bit further to tell you about how we can use top-down proteomics in this top-down overview of our samples to look at genetic and alternative splicing isoforms together with post-translational modifications, which is actually a big difference between this top-down method and the conventionally used bottom-up proteomics that people might be more familiar with. So in bottom-up proteomics, what we do is we take our proteins and we proteolytically digest them prior to mass spec analysis. And what this does is we actually lose a lot of information. We never actually observe the intact proteoforms. And so this means that we might lose information about what isoform PTM combination we might actually have. And when we're measuring those in our mass spectra, we really want to be able to measure the intact proteoform peak to make accurate comparisons. So that is one big difference of this top-down proteomics method versus the conventional, more familiar bottom-up proteomics method. So here you can see I'm just showing you an example of tropomyosin. The most abundant isoform that we observed in our samples was TPM 1.1, and it's also commonly referred to as alpha tropomyosin. You can see that we see the unphosphorylated or unmodified form, and we also see a phosphorylated form. But if you were to zoom into the baseline here, you can actually see much more. So you can see an alternatively spliced isoform of TPM1, so we call it TPM 1.2. We also see isoforms from different genes. So we can see TPM 3.12, which is also gamma tropomyosin. And you can also see beta tropomyosin, which comes from gene TP2. And then in addition to these genetic isoforms that we're able to see from tropomyosin, we can also see short cipher isoforms. So this is spliced from the same gene, LDB3. So you can see cipher 6 in its phosphorylated form, and you can also see cipher 5, phosphorylated cipher 5, and bisphosphorylated cipher 5. So you can get a lot of information by using this technique. And if you were to digest these proteins, you might actually lose information about where the signal is actually coming from and where the phosphorylation is coming from. Okay, so what we then wanted to do after we went through and identified these proteins and saw what we could look at in our sample, we wanted to make quantitative comparisons between the donor tissues and the HCM tissues and how the proteoforms are changing in those samples. So first we looked at cardiac troponin I. So you can see that we can see the unmodified cardiac troponin I, monophosphorylated cardiac troponin I, and bisphosphorylated cardiac troponin I as our three major proteoforms for this protein. And you can see that we know they're phosphorylated because of the mass shift we see between the peaks. So this indicates a phosphorylation of 79.96 Daltons. And you can see 3 representative spectra from the control and 3 representative spectra from the HCM samples. So you can see actually that in the HCM samples, you see this decrease in phosphorylation. So you see an increase of the unmodified version of the protein. So that indicates a decrease. And so this isn't anything new. We have, other groups have observed that there's a decrease in phosphorylation in tissue from HCM patients. So this is not a new finding, but what was a new finding was when we looked at ENH2, which is a Z-disc protein, it comes from the gene PDLIM5. We could also see 3 different proteoforms for this protein. So you see the unmodified, phosphorylated, and bisphosphorylated form. And you also see this decrease in phosphorylation in the HCM samples. So that was really interesting. So when we, and additionally, this is just regardless of what the mutation was. So you see this change in proteoform profile. It's pretty consistent regardless of the genetic mutation that causes the disease for that patient. And you can see this consistently for the whole cohort for the HCM patients. You can see that there's a really consistent decrease in this phosphorylation for these 2 proteins. And then I'm just showing you now all 8 samples that we have genetic information for, and you see that still, regardless of the genotype, we can see this really consistent proteoform profile for these 2 proteins. And it's remarkably consistent. And also I wanted to indicate here that you can see polymorphisms in the proteoform profile that we see here. Okay, so we noticed this consistent decrease. So we also wanted to know, is there like a correlation between the cardiac troponin I and ENH2 phosphorylation? So what we did was we mapped the phosphorylation for these 2 proteoforms, or these 2 proteins against each other, and we actually saw a really positive linear correlation between those samples. So that kind of told us maybe these are being regulated by a similar kinase, or maybe there's crosstalk between the pathways that regulate these proteins. And so we really wanted to answer that question. And what we did was we started by mapping the phosphorylation sites on the proteoforms. So first, we used tandem MS, which is MS. And that allows us to take the proteoform and get sequence information about it. So we can obtain amino acid sequence information. We can also localize the phosphorylation sites. So when we did this for cardiac troponin I, we mapped the phosphorylation sites to serine 22 and serine 23, which is a consensus pKa site. So that is something that's pretty well known. And then we were able to map the phosphorylation site for Enigma homolog 2 to serine I believe it's 118. And you can actually see here that there's a consensus pKa phosphorylation site here. So this indicates that potentially these proteins are both regulated by pKa, and maybe there's a dysregulation in pKa signaling in the HCM samples. Okay, and then moving on to another member of the troponin complex in the sarcomere, cardiac troponin T, we actually observed that there was an increase, a slight increase in the phosphorylation for cardiac troponin T in the HCM samples. Our collaborator, Steve Marston's group, actually a few years back found that troponin, or TNI phosphorylation, is uncoupled with myofibular calcium sensitivity in HCM hearts. And they actually were able to link this to a functional abnormality in cardiac troponin T. So what they did was they replaced the endogenous cardiac troponin T with a recombinant TNT, and they were actually able to recover the coupling of phosphorylation of cardiac troponin I and calcium sensitivity. So they then linked this functional abnormality to TNT, but they weren't able to actually determine the molecular cause of this. But we actually show here that there seems to be, while very slight, an increase in cardiac troponin T phosphorylation in our HCM samples compared to donor samples. So this indicates that potentially this could be one of the links to investigate for what Steve Marston's group has found. And then just to talk more about these cipher isoforms that we were able to detect. So I mentioned before that these all come from the same gene, LDB3, and it's a gene that's actually expressed in striated muscle tissue, and it exists in many splicing isoforms. And these are the 2 short isoforms that do not contain the limb domain. And they're not really well understood what the function of these isoforms are, so they haven't been previously characterized, but you can see actually that there seems to be a slight increase in cipher 5 phosphorylation in HCM samples. However, they were so low abundance that we actually couldn't reliably quantify this, but just as a qualitative observation, you can see that you start to see a tetraphosphorylated form of cipher 5 arising here from the baseline in these HCM samples. So we really wanted to know where the phosphorylation sites for cipher 5 and cipher 6 were, so that we could sort of, in the similar way to cardiac troponin I and ENH2 that I showed in a few slides earlier, understand what sites were being phosphorylated so that we could start to know maybe what kinases are acting on these proteins and understand why we might see this dysregulation. Unfortunately, because the phosphorylation sites are so low, we weren't able to actually map the phosphorylations for these proteins, but we're able to characterize the sequences from endogenous human tissue for these proteins for the first time. So we actually got pretty nice sequence coverage for both of these proteins. And if you take a close look, you can actually see that the sequences for these are actually really close. And so by top-down proteomics, this is a unique piece of information that we can get, because if we were to use bottom-up proteomics to characterize these proteins, we might be mixing peptides because they are so high in sequence homology. So just to summarize, I didn't show you all of the results of this analysis of donor heart tissue with HCM heart tissue, but just to summarize, we're able to take a look at a complex sarcomere proteoform landscape using top-down proteomics. In addition, we saw a decrease in cardiac troponin I, ENH2, TPM 1.1, and MLC2V phosphorylation. So that's a myosin light chain to ventricular isoform in our HCM samples. And in addition to that, we saw an increase in cardiac troponin T phosphorylation in HCM, which we potentially could investigate as a link to the uncoupling of calcium sensitivity and cardiac troponin I phosphorylation in the sarcomere. In addition, this convergence at the proteoform level seems to be independent of HCM genotype. So if we're thinking back to that initial question that we posed, how is genotype related to this proteoform profile and phenotype, and how can these converge to highly similar phenotypes? And we're seeing actually that there's a remarkably consistent convergence in the proteoform profiles that we're seeing in the HCM samples compared to the control samples. And this might indicate a common pathway is activated in HCM, or common pathways, and potentially present opportunities to develop therapeutics, similar therapeutics across the board, instead of just based on the genotype that we see for the patients. Okay, and so up until now, I've shown you how we can use top-down proteomics to look at just the sarcomeric subproteome, but we know that there's actually a lot more going on in our heart than just what's happening in the sarcomere. So we have a lot of group members who are also working on top-down proteomics of membrane proteins, phosphoproteins, and the kinome. And I'm actually going to talk to you now a little bit about some technology development for this platform that we're using to study heart tissue and heart proteoforms. So just to remind you what you might see if we were to use just one dimension of liquid chromatography separation for a mixture of heart tissue proteins, or proteins extracted from heart tissue. So you can actually see here, this is just a 1D or one dimension reverse phase chromatogram, and you can see members of the troponin complex. So you see cardiac troponin I, cardiac troponin T, and troponin C. And you can also see the myosin light chains, so myosin light chain 1 and myosin light chain 2, and then in addition, cardiac alpha actin. And I'm showing you the mass spectra here. This is on an M/Z axis. But going to a mass axis, like what you're familiar with looking at, you can see a lot of proteoforms for these proteins, as I mentioned in previous slides. And this allows us to compare and quantify their abundances between samples. However, one thing I want you to notice is that most of these are actually just highly abundant proteins in our hearts. So we know that the sarcomere proteins are pretty high abundance, especially the myofilament proteins. And then most of what we can detect are below 30 kilodaltons in molecular weight. So this is actually a pretty common problem for top-down proteomics, and why we need to further develop top-down proteomics methods to be able to access higher molecular weight proteins in the heart and do the similar quantitation that I was showing you in previous slides for those proteins as well. So just to repeat myself a little bit here, you can see an interactome of the sarcomere, and you can see what we've identified, as I mentioned, has mostly been proteins less than 30 kilodaltons and especially less than 60 kilodaltons. But we know that we actually have a lot more in our heart that are greater than 60 kilodaltons in size, and especially myosin heavy chain, which is 223 kilodaltons, and cardiac myosin binding protein C, which is 140 kilodaltons. And we know those proteins are really important. And then there's also proteins in our Z disk which is a signaling hotspot in the sarcomere that we really want to be able to analyze and look at the different proteoforms that are arising. So a lot of my doctoral work actually has focused on method development toward top-down proteomic analysis of our heart proteome. And it's, as we know, highly complex and also has a really high dynamic range, which are both challenges for top-down proteomics. And in addition, we really care about looking at larger proteoforms in the heart, but that's also a challenge for top-down proteomics, as I'll mention on the next slide. But we've been posing solutions such as using multiple dimensions of liquid chromatography separation for complex mixtures of proteins, and then also size-based fractionation at the front end of our top-down proteomic platforms to solve some of these issues that I mentioned. So we know that mass spec detection of large proteins is pretty challenging, and I'll tell you why that is. If you look at this chart here, it actually shows you really nicely. If you look at the x-axis, which is mass, as protein mass increases, our mass spec signal to noise actually exponentially decreases. So that's one problem related to the mass spec detection of large proteins in general. So if you combine this issue with the coelution of low molecular weight proteins and also high abundance proteins like the myofilament proteins, we're going to see detrimental signal suppression of the high molecular weight proteoforms that we're interested in. So one way we can get around that issue is by doing a size-based separation prior to reverse phase chromatography separation. And one way we can do this is by doing size exclusion chromatography, it was previously thought to be pretty low resolution, but we actually showed that with the right combination of pore sizes, we could actually get really nice size-based separation of our complex mixture of heart proteins extracted from tissue. So you can see here, these are just a gel, this is just a gel representation of the proteins that we're able to fractionate by size. And then we could take these fractions and further analyze them by reverse phase chromatography, which is what I'm showing you on this slide. So you can see now that we were able to separate those high abundance, low molecular weight myofilament proteins from the larger proteins, and we're able to enrich for those larger proteins. And overall, we were able to expand the coverage of our heart proteoform landscape, or what we could detect by top-down proteomics. But really importantly, we were able to increase the number of proteoforms greater than 60 kilodaltons that we could detect with mass spec at this point. So just showing you one example, and this looks like a lot of noise, but I'll tell you that it's a 140 kilodalton protein, and we were interested in identifying it. So we did tandem mass spectrometry, or MS2, and we got this really complex MS2 spectrum, but we were able to piece it back together with using some software tools. And we were able to identify it as cardiac myosin binding protein C, which is actually one of the important proteins that I mentioned earlier in the interactome. So we were now able to use this technology to enable us to detect these larger proteoforms in the heart. And we really care about this protein because mutations and altered PTM states have been implicated in heart diseases, including HCM, which I talked about in the earlier part of this talk. So now we've expanded on the coverage of the sarcomere proteoform landscape using this technology. We were able to also detect myosin heavy chain, 223 kilodaltons. And then additionally, we were able to detect nebulate and multiple phosphorylated proteoforms of nebulate, which indicates that it's probably involved in some sort of signaling in the sarcomere. So just to summarize, I've shown you that top-down proteomics has revealed distinct HCM genotypes actually converge to similar proteoform profiles in septal myectomy tissues from HCM patients compared to donor heart tissues. And additionally, we've used an MS-compatible SEC fractionation technique at the front end of our conventional top-down proteomic platforms to increase proteome coverage of the heart, especially for those that are greater than 60 kilodaltons. And so we've used this technology to expand on our heart proteoform catalog. And this will allow us in the future to hopefully use this to make quantitative comparisons in healthy and diseased samples. However, there's still some technological challenges associated with this later method that I talked to you about, the serial size exclusion method. It actually required quite a bit of tissue. So we really want to scale this down so that we could start to analyze samples closer to what we had available for our HCM study, which is just very small amounts of tissue. So we really want to increase top-down proteomic sensitivity and develop this method further so that we can start to look at smaller amounts of sample and make these comparisons between large proteoforms in the heart. And then in addition, a lot of what I showed you today comes from tissue. But we know that tissue is not easy to come by, especially from healthy hearts. And also, we are only able to get a snapshot of HCM at the end stage of the disease if we're analyzing tissue that is taken from patients who go in for septal myectomy procedures. And so if we could model this disease in a cell-based model using induced pluripotent stem cells, we could get a snapshot of HCM at different points, at different time points during the disease progression. And so that is the goal. As I mentioned earlier, I worked closely during my PhD with Dr. Wenchuan Kai, and this is some of her work that I'm showing you, but she actually has developed a platform using top-down proteomics to assess the maturation of human-induced pluripotent stem cell-based cardiomyocytes. And we hope to use this method to further develop these cardiomyocyte-based models for HCM so that we can start to get a better idea of the disease progression over time rather than just at the endpoint. And we're hoping that this can also be used to model other different diseases in the heart as well. And so with that, I really want to acknowledge my mentor, Professor Inga. She's really sad that she couldn't be here today, of course, but I would like to acknowledge all of the advice and mentoring that she's given me over the years, and all of the group members who have been involved in collecting this data and performing this research are listed here. Specifically, I really want to shout out to Dr. Wenchuan Kai, who's my co-first author on the HCM work that I showed you, and also a co-first author on the SEC work. So we've actually worked pretty closely. And Dr. Zach Gregorich in our group has been instrumental in the HCM project as well. And then we also, as I mentioned, have many collaborators, international and in the US, that have helped with tissue procurement and then also talked with us through a lot of the things that we're seeing happening in the sarcomere proteome. So, and then also our funding. And I'd like to thank you for your attention. And we're also on Twitter if you are on Twitter. So feel free to follow us and follow up with me if you have questions. And with that, I'd like to take any questions.
Thank you very much, Trisha. That was a great talk. I learned a lot. So we've got quite a few questions already. Maybe I can just start with one while people are still writing their questions. Probably a really simple question, but I'm just interested in the heterogeneity you're obviously finding as a proteoforms. It includes post-translational modifications or polymorphisms, so all the different forms of the proteins. Do you see in the control samples compared to the HCM, the heterogeneity, is that greater or less? I'm wondering if it's actually greater in the control. There might be a lot of sort of benign or even beneficial morphisms in the control compared to the disease. I was just wondering.
Yeah, that's a really great question. And so to sort of, I think there's maybe two parts to this. So to address the heterogeneity of the donor tissues, you can see that actually there's a wider spread in what we were able to see in terms of like phosphorylation of certain proteins. And we think that has a lot to do with differences in geographic location, comorbidities that might be involved in, the donor tissues we were obtaining. And then also our HCM tissues were collected in a very uniform way, whereas our donor tissues, while we did the best we could to, collect those and preserve what we're seeing in the heart at the time that the tissue is collected, there's obviously a lot of differences that can happen there. But in terms of answering your question about polymorphisms, we see, across these samples, so I looked at 32 different samples, you could see for a few genes, you could probably see about like 3 or 4 polymorphisms. Some are the same, some are different. I think it just has a lot to do with, the frequency of a specific polymorphism in the population. But does that answer your question or start to answer your question?
Yeah, I think so. Yeah, thank you.
Mm-hmm.
Good. So maybe we can get started on some of the audience questions. I think quite a lot of them are technical.
Sure,
so I'll start off with the first question from Trayambak Bazak, who asks, how do you take care of complex eluting as a single peak in chromatogram?
Oh, sure. So I believe the question is about whether or not we can look at protein complexes versus individual proteins. So for this specific analysis, it's all denatured. So everything is actually denatured. So we're assuming that nothing is in its complex form. But there is a technique that is sort of similar to this. It's called native top-down or native MS, that we can use more native conditions. So just to give you an example, we're using pretty harsh denaturing conditions, not harsh, but 1% formic acid in water is used during the SEC fractionation. So that should, in theory, break any interactions between the proteins so that we can get each individual protein to elute as a monomer. But there are techniques used or available to look at native complexes and how they elute, and that's a developing field as well.
Okay, thank you. The next question is from Monica Gladka. Hi, Monica. She says, very nice talk. How do you extract sarcomeric proteins from the heart? Is there a specific protocol? I was also wondering, actually.
I'm sorry, could you repeat that question?
How do you extract sarcomeric proteins specifically from the heart? You mentioned that's your first step from the tissue.
Yes. So there's actually a protocol that you can use to specifically, so a lot of these sarcomere proteins are acid soluble. So what we do first is we homogenize the tissue and we extract the cytosolic proteins in like a pH 7 solution. We just use heap stuffer. So that extracts all of the cytosolic proteins, and then we can enrich for the sarcomeric proteins using, we use a 1% TFA extract, and we extract all of the acid soluble sarcomeric proteins in that fraction. And so we can enrich for them that way. There's also another way that's more native. You can use a 0.7 molar lithium chloride extraction to specifically extract the sarcomere fraction.
Thank you. I'm making notes.
Sure. If you need the protocol, I can happily send it to you.
Trisha, do you mind if I just ask a follow-up question to that? So sure. if you were, for example, advising people such as me who run Western blots and obviously knows very little about proteomics, how to kind of get the most proteins in your solution, what would be your advice actually?
Yeah, so the most
proteins.
Sure. So in terms of extracting for Western blot, I would say people tend to do a whole cell extract using a detergent. And so you can use detergents to just extract the whole cell lysate. And I think a lot of people do that for their Western blot analysis. So they will just do a whole cell lysate extract. And then that hopefully gives you the most proteins in your lane for a Western blot. You can also do, I suppose might work a little bit better to do even just fractionation of different parts and then do separate lanes for your Western blot. But that it's the same concept would apply for mass spec, is that if you fractionate at the level of extraction, when you're extracting from your tissue or your cells, you're able to see more because you're enriching for certain proteins in different fractions. And so that's actually one of the better ways to do it for at least mass spec as well. The one thing that I'll say, because I mentioned detergent is, at least SDS and a lot of other ionic detergents are very incompatible with MS, but there are a lot of available MS compatible detergents, and there's one specifically that our group just developed that is a photocleavable surfactant. So you use it to extract all of your proteins, and then you can just use UV light to get rid of all of the MS incompatible parts before that. So there are a lot of options for those things too.
Okay, great, thank you. Next question is from Viba Rani, who's, I think they wrote a question earlier and then got something wrong. Do you add protease inhibitors in isolated samples?
Yes, yes. So at the extraction level, there are protease inhibitors, there are phosphatase inhibitors. There's a cocktail that we use to inhibit for different proteases and phosphatases to ensure that what we are extracting is as close to the native PTMs that we want to see as possible. So there are those that we add into our mixture.
Thank you. The next question is from Danuta Cesna Cordery. Sorry if I pronounced that badly. Says, greetings from Miami. Have you looked at the phosphorylation of MLC2 in HCM sample versus donor septal tissue?
Yeah, MLC2, is that the protein? Yes, yes. So actually that was one of the proteins that we found to have changing phosphorylation. So it actually decreases, we found, in the HCM samples compared to the donor tissues.
Okay, thank you.
May I just butt in? Danuta has a postdoc position for anybody that's listening, so I would highly encourage you to apply.
Okay, great. So the next question is from an anonymous attendee who asks, as a technical question, is the mass of 18 to 30 kilodaltons restrictive for this method? I guess you discussed that at the end.
Yeah, so I would say it just depends what platform you're using. I would say pretty commonly now you can get up to 30 kilodaltons easily using top-down proteomics. If you go a little bit further using a size-based fractionation technique, you can definitely get deeper into the proteome and then looking at larger proteins. But it's not, I would say, as of now, conventionally looking at larger proteoforms using this technique is not considered conventional, but I would say things below 30 kilodaltons would be pretty easy.
Okay, thank you. The next question is from Lucy Carrier, and she says, hi Trisha, beautiful approach. I guess this is a comment, so please note that MYBPC3, the E542Q mutation, is actually not a missense, but induces skipping of exon 17 and a frame shift with a truncation in C4.
That's good to know. Thank you.
Okay. The next question is from Junhei Sun, who says, with the multiple or selective reaction monitoring, MRM or SRM, hopefully that makes sense to you, proteomics, helpful for protein quantification using a surrogate specific peptide?
So that's a really good question. So we didn't use SRM or MRM in this technique, and that's just a way to select for a specific ion or M over Z in the mass spectrum. And people use that a lot for quantitation for bottom-up proteomics. We haven't really used this technique for our top-down method. For us, a lot of the proteins we look at, we already kind of know what they are based on their mass and past MSMS confirmation of their sequence. So in a lot of ways, we just use the intact mass intensity for the quantification. So we do relative quantification of different proteoforms within a sample, and then we can compare those relative quantified values between samples.
Yeah, okay, thank you very much. Another question from Danuta asks, does PP troponin I, so PPTNI, phosphorylated depict phosphorylation at two adjacent sites, serine 32 and 33, or are there phosphorylated residues elsewhere in the troponin I molecule?
Right, so the MSMS data that we collected indicate, and from past studies that we've done about cardiac troponin I phosphorylation, we've found that they are sequential phosphorylation events. I can't remember which one comes first, but one of them comes first. We have confirmed in the monophosphorylated proteoform that there is one phosphorylation site, and then in the bisphosphorylated form there are two, and so that indicates that there are sequential phosphorylation events that are happening there. So we can get that information from the MSMS data as well.
Okay, thank you. The next question is from Konstantina Stathopoulou, who says, interesting talk. Could you also detect oxidative post-translational modifications in sarcomeric proteins in your experiments? Yes,
absolutely. So the oxidation would be just a 16 Dalton shift from the unmodified peak or any other peak. So we can absolutely detect that. And so depending on how high resolving power your mass spectrometer is, you can actually detect things even 1 Dalton away from the proteoform peak. So you can detect things like deamidation even. So there are really the only limit you have there is your resolution on your instrument. So, but for this specifically, yes, we can detect oxidation as well.
Great, thank you. Sina Hadipur Lakamasara asks, says, great to talk, Trisha. I have a few questions. So question 1 first, are these healthy donor heart tissue, and what comorbidities did they have? You mentioned.
Yeah, right. So we only have certain information about the donor heart tissues. That just depends where we got them from and how much information we were provided at the time that we obtained the samples. But we do have some information about family history. So we wanted to ensure that these samples didn't have a history of heart disease, because that was really important if we're trying to, treat these as sort of controls to compare against our HCM samples. So that was important to us. So that's information that we had. And then we did have cause of death for some of them. So for most of them, I would say we had cause of death information. And then we did, I believe, have some information about the comorbidities, but we importantly just confirmed that they didn't have a history of heart disease. And so that's about the extent that we went into looking at that.
Okay, I'm going to ask, were they age matched? Do you know that roughly?
So we didn't do age matching. We just had a cohort of different ages. I think the youngest in our donor was 14. I don't know the oldest. For the HCM, I think most of them were in their 30s, 40s.
Yeah.
When the tissue was collected.
Okay, thank you. And then next question from Sina says, do you think that the level of phosphorylated cardiac troponin I, ENH2, TPM 1.1 and MLC2V protein has decreased, or is it possible that due to hypertrophy, the increased expression of proteins are hiding the signal of phosphorylated protein? Because I guess it's relative. Yeah.
Oh, yeah. So everything's relative, but since the unmodified proteoform coelutes with the phosphorylated proteoform, we can measure them in the same snapshot of a spectrum. And so everything that we detect comes from, how do I put this? We also do a relative amount of the total protein that we see. So for example, when we do our quantitation, if we were to measure phosphorylation of, let's say, MLC2V, we would take the phosphorylation intensity over the total MLC that we see, and that allows us to adjust for any MLC amount differences in the samples. Does that answer the question? If not, please feel free to follow up with another question.
Yeah, I guess I think that probably does answer it. And let us know if it doesn't, Sina. And the third part is, have you correlated specific phosphorylation changes to severity of disease in HCM group?
That's a really good question. So in terms of severity, so this isn't quite my expertise, of course. So if there's somebody who has more information about this and can follow up, that would be great. But we collected all of the HCM tissues at the point where the patient went in for a septal myectomy. And so that means that there was an outflow obstruction. And so at the point we obtained the HCM tissues, they were all presumably at the point where they needed a septal myectomy procedure to relieve their symptoms. And so we believe that this means that we're sampling the disease at about the same severity, I would say. But I think that there's probably, as I mentioned in the later slide, we really want to take a look at this disease as it progresses in the cell-based models, and that would give us a lot more time-resolved information and severity-resolved information, I think, about what's happening here.
Yeah, thank you. The next question from Conrad Winkles says, great talk. Do you see retention time shifts in regard to degree of phosphorylation, or do they coelute with non-phosphorylated proteoforms?
Yeah, that's a great question. So in terms of their elution times and their hydrophobicities, that So their elution times are going to be based on the hydrophobicity differences. And we've seen that for intact proteins, phosphorylation moieties are pretty small, and while they're charged, they don't really affect the elution time of the protein very much. And so when we're doing our quantification, we make sure that we take into account the whole elution profile for that specific protein, but we found that the phosphorylated proteoforms actually elute within seconds of the unmodified version of the protein. And that would obviously change based on what type of reverse phase chromatography you're using and things like that. But in general, we found that the phosphorylation moiety doesn't really affect the hydrophobicity and the elution time very much. So they can be quantified within the same spectrum.
Okay, thank you. The next question from Leslie Kennedy asks, for alternative splicing events, since proteomic analysis typically utilizes database matches for protein identification, wouldn't alternatively spliced variants be missed if they haven't been annotated in a database? This is in comparison to RNA-seq, where you get sort of unbiased sequence information for identifying novel variants.
Yes, absolutely. That's a great question. Yeah, so we're limited by our databases. Absolutely. So if there's something that we don't know about in terms of like an alternatively spliced variant, we may not be able to identify it. We can definitely use mass spec to get pretty close. So what we can do is we can look at the intact mass, and we can do tandem mass spectrometry to figure out its sequence. And that's an ideal situation that you get really nice sequence coverage with tandem MS. But ideally, in my opinion, I think that transcriptomics and genomics and proteomics should all be combined to kind of help us figure out what these things are. And I definitely agree that transcriptomics is a great way to try to build our databases and make them more comprehensive.
Multi-omics.
Mm-hmm.
Yeah. Another question from Monica Gladka. She says, non-failing tissue was collected from left ventricle and HCM was from the septum. Do you think the differences in phosphorylation status and sarcomere proteins are purely because of the location in the heart?
Yeah, that's a great question. That's something that we actually thought about because we were, we're like, oh, we don't want to compare apples to oranges. So we actually did a comparison in donor heart tissue between the septum and the LV, and we confirmed that there weren't differences between the proteoform profiles in the septum and the LV. And actually somebody in my group, Elizabeth, she's actually working to look at 11 different regions of the heart and compare those proteoform profiles to ensure that what we are comparing is comparable.
Okay. Thank you.
Emma, sorry, if you don't mind me just asking, I mean, you're obviously very familiar with the kind of genomic assessment of the heart and different parts of the heart. That's probably not the case, really. If you're looking at, let's say, genomic profiles from different hearts, well, from different parts of the heart, they're probably fairly different, are they not? Or do you still see?
Well, the genomic profile will be the same,
yes,
but the transcriptomic is different, yeah.
Okay, so that's what I mean. So
yeah. Well, in the RNA sequencing, so to be fair, we haven't sequenced septum differently to other parts. It tends to be normally with the left ventricle or just cut out altogether, but certainly in different chambers, there's differences in the transcriptome, yeah.
Or the epigenetic changes that are they different
Well,
in different
how
parts?
the transcriptome is different is because of the epigenetic differences, I guess, different parts of the heart, different stressors, and yeah.
Okay.
So it's
forgive me for my naivety.
yeah, but yeah, it's good to have a multi-omics approach, like you say, and overlay everything. And
absolutely.
Yeah. Great. Another question from Leslie Kennedy asks, can you speak a little bit about the physiological impact of the ENH2 phosphorylation changes impacting cardiac troponin TI phosphorylation?
That's a really great question. I cannot say that I know exactly what ENH2's role is. I don't think I did a really extensive literature search into this. We don't really understand what this shorter isoform of Enigma homolog, what its role is in the heart. We know that the longer version of this protein, which has a limb domain, is localized to the Z disc. And I believe, I don't want to say something incorrect. I'll leave that because I don't want to say anything incorrect, but we don't know what the shorter isoform's role is specifically, but we know that it localizes to the Z disc. And so, yeah, there's a lot still to be learned there. We kind of hypothesized that maybe cardiac troponin I and ENH2, since we've shown that they are likely phosphorylated by pKa, and the changes are related to pKa differences or dysregulation. So they might not be acting together in any way, but maybe there's some type of common pathway that is dysregulated, and these are signatures of that dysregulation. And then as a result, somehow something in the sarcomere is not working as it should.
Yeah. Okay, thank you. Next question from Bruce. Are you still there, Trisha? Or is my internet gone? Can you hear?
No, I think you're fine. Trisha's just frozen. She's still here, but
give it a moment.
Hopefully it'll come back. If not, she can just rejoin. But
make sure we keep the questions as well.
Yes, we apologize for this.
Yeah.
Hopefully Trisha can rejoin us soon.
Big distance.
Oh, there we go. She's rejoining.
There we go.
Great. So yes, make sure I'm in the right place. I've just scrolling down.
It's Bruce Allen next,
yeah,
right?
Thank you. Yeah, next question from Bruce Allen. For your size exclusion chromatography, are you using chaotrope before or during the run to dissociate protein complexes?
Yeah, that's a great question. So as I mentioned earlier, it's really strongly denaturing, so it should, in theory, break all of the complex interactions. But we do use a little bit of hexafluoroisopropanol, HFIP, which is a chaotropic agent that should break stronger interactions that maybe our formic acid in water isn't able to break. And then later down the line, when we do our reverse phase chromatography, we also use an organic solvent in our elution. And so that should also further break up anything that is in complexes.
Okay, thank you. Trisha, you're happy to carry on with the questions?
Yeah, sure, absolutely.
Okay, yeah, there's quite a few more.
Sure.
So Miles Housley asks, could you use this method for low abundance high molecular weight, say 1 to 200 kilodaltons, by first immunopurifying and then subsequent analysis?
Yeah. So as I was mentioning earlier about, enriching for certain things, any type of enrichment that you do definitely helps your proteomic analysis, especially if you want to look at very specific things. And so people actually do and have done top-down proteomic analysis of purified proteins as well, and that actually helps you when you're trying to do really extensive tandem MS and localized phosphorylation sites and things like that. So yeah, you can certainly look at lower abundance proteins if you do an enrichment. It definitely, I think, like any enrichment, if you're going to try to look at larger proteins, it has to be pretty clean, because remember that anything that you pull down that is lower molecular weight will also need to be separated at least by, reverse phase chromatography or something else before you can see the signal from the larger proteins. But in an ideal case, if you were able to pull down a lower abundance, high molecular weight protein, you should be able to see it.
Okay, thank you. Sila Algul says, hi, and thank you for your interesting talk. Are you planning to look at proteomic differences between asymptomatic mutation carriers and symptomatic mutation carriers?
That's a really great question. So there is work coming down the line. I'm not as familiar with this part of the research, the grant that was written for this, but what they're planning to do is look at a family carrying an HCM mutation, so a single gene mutation, and they have members of the family who present clinically for the disease and who don't. And I believe they're taking those different cell lines and differentiating them to do the induced pluripotent stem cell models for the disease, and seeing how, age affects things, or gender, just different people.
And they might have other polymorphisms that are beneficial, that sort of somehow override the...
Yeah. Definitely. And I think environmental factors also be not within a family, I suppose, but maybe even within a family. But I hear that environmental factors makes a difference as well for, whether somebody might present for the disease or not.
epigenetics.
Yes, of course.
So Anat Kopra asks, do you have any examples in your data set where on HCM where you do see clear differences in protein phosphorylation pattern between mutations? You said mostly it seemed to be independent of the mutation, but were there any examples where there were differences?
Yeah. And so for the most part, we saw very consistent profiles. The exception was for things that were lower abundance. We sometimes could not tell whether there were differences due to just having a really low abundance proteoform that we just maybe couldn't detect in one sample versus another, and that gives us a difference. So we tried the best we could to exclude anything that we couldn't reliably quantify from this analysis. However, there was an example in our tropomyosin that we actually, in all of the donor tissues, we didn't see gamma tropomyosin. So in 0 of the donor tissues, we were able to detect gamma tropomyosin. However, in 8 of the HCM samples, we were able to detect gamma tropomyosin. So that doesn't mean that it's not in the donor samples. It just means that it was below our detection limit, of course. But for 8 of them, we were able to detect that protein. So that seems to be a difference, but we weren't able to reliably quantify that because of how low abundance it was. So it could be a difference. We weren't able to look at the genotype versus those samples that had gamma tropomyosin and didn't, and find a correlation, because most of the samples that we had here were myosin heavy chain or myosin binding protein C mutations. But that would be really interesting to look into to see if that is a true difference. But for the most part, we've seen everything converge, which is really interesting.
Okay, thank you. Another question from Bruce Allen asks, when considering the post-translational modifications of proteins, do you take into account possible or medication that the patients are taking?
Yeah. So we have a list of, for most of the HCM samples, we have a list of the medications they were on, and we definitely recognize that, medications and comorbidities and things like that could affect the proteoform profiles. But as I mentioned, the HCM profiles that we saw were very consistent, while patients were on different medications, different lists of medications. So that is provided in once this is out into the world, we have that information in our SI for this.
Okay, thank you. Beta or Wolska says, very nice talk. Cardiac troponin has multiple phosphorylation sites. Are you seeing more sites? I don't know if they mean more sites than are previously described, I guess.
Mm-hmm. Yeah, so the major proteoforms that we see for cardiac troponin I, we only see a monophosphorylation and a bisphosphorylation. So those are the major proteoforms we see. Presumably there could obviously, since there are more phosphorylation sites on this protein, we could be just not seeing lower abundance forms. That's definitely a possibility. That's something that we could presumably use bottom-up proteomics to answer that question, because it can detect lower abundance phosphorylation sites in addition to the major phosphorylation sites that we're seeing. But the ones that we're seeing that are major, we've detected as serine 23 and 22.
Okay.
But just to sort of butt in a bit, I mean, combining Bruce's and Beata's question, if you, for example, were to look at a patient on beta blockers, would you distinguish that patient? I know it's a complex thing because beta blockade doesn't necessarily always block phosphorylation, because it's probably something strange tends to happen maybe physiologically, well, in cells. But do you see any differences between those patients on beta blockade and the ones that are not on them? Because that to me seemed like something that you could pick up, right?
Right. Yeah, no, that's a really great question. It's something that we've been talking about as well, whether or not these phosphorylation profiles are affected by the patients being on beta blockers or not. That's something we haven't correlated yet. So we haven't taken those samples that were on beta blockers and versus the ones that weren't and looked at the differences, because we've seen such a consistent change. And so we could definitely, I think that's a really great idea. We could take and look at those differences. I will mention that in a previous paper by our group that looked at heart failure, different samples from different patients that were at the stage of heart failure, and then I believe also dilated cardiomyopathy and ischemic cardiomyopathy, they did that correlation of the patients that were on beta blockers versus not, and they were not able to find a difference in the cardiac troponin I. It's really interesting. Yeah, no, I'm happy to send you that paper. But they did, because we were preparing for these questions, I knew that someone was going to ask about the beta blockers, and we've been kind of racking our brain, like, is it related, is it not? And it's definitely possible, but we haven't seen a correlation, at least in that previous study, and we can certainly look here, but we do see really consistent.
That I think poses even more questions about the role of beta blockers in treatment of heart failure and generally cardiovascular disease, doesn't it?
Right, yes, absolutely.
It's not changing the molecular phenotype. Yeah, we've had trouble, actually. I was hoping to do a similar thing with a recent study. We had trouble finding a disease tissue where the patients weren't on beta blockers and ACE inhibitors. Like, a lot of them are on
right.
those.
It's hard to really like break apart what those effects from the drugs are to the actual proteoform profiles, and I think that's hopefully what we can start to do with these cell-based models. Not actually taking tissue, but really just looking at a really controlled system and seeing what the effect truly is.
Yeah, thank you. Back to the audience questions. Next question is from Kashiki Mahapatra, and they ask, is the extraction protocol different or less harsh between heart tissue versus cardiomyocytes generated from iPS lines?
Yeah, that's a really great question. So I haven't personally worked with the cardiomyocyte extractions, but for the tissues, we just use a handheld homogenizer, and we, do our classic cytosolic extraction and then an acid-based sarcomere extraction. And I would imagine that for cells, it wouldn't be very much different, because I think just really the important thing is actually breaking the cells and releasing the protein so that we can enrich for them.
Yeah. I imagine it would be less harsh as well, yeah. The next question is from, I apologize about the pronunciation, Christoph, yeah.
Call him Chris, don't worry.
Christoph, yeah. Well, and he asks, is there a possibility to characterize the hydrophobic PTMs like lipidation during the top-down proteomics?
Yeah, that's an awesome question. Yeah, and so I would imagine for things like lipids or lipid modifications, things that are a little more hydrophobic, that you might see a slight difference in retention time, going back to an earlier question about the phosphorylation. And I think that has been the case. A few members of our group have looked at palmitylation, actually, which is a lipid modification for, I think, phospholambin is the protein they're looking at. It's smaller, so of course, I think the smaller the protein, the difference between the PTM size is going to have more of an impact. But yeah, we definitely can look at those types of modifications. And there are also, for membrane proteins, other types of separation methods that are used to kind of take advantage of the differences of the hydrophobicity for those proteins and their modifications too. So there's something called HILIC, which is just the opposite of reverse phase chromatography. It separates by, hydrophilicity differences.
Great, thank you. Making more notes.
Yeah.
Next question from Rio Juni. Hi, Rio. It says, great work. Some people have sarcomeric mutations but do not manifest the disease, so asymptomatic. Do you perhaps have these samples as well, and whether you see a difference in proteoforms compared to samples from the symptomatic HCM patients?
Yeah, that's a great question. So one of the challenges, I think, for this type of analysis is our access to the tissues. And so the reason we have access to these HCM tissues in the first place is because these patients were symptomatic and they needed to undergo the septal myectomy to relieve their symptoms. So that's why we have these samples specifically. And so getting tissues from somebody who might not have the disease presentation, but they have a mutation, is pretty challenging. Because as you can imagine, a healthy person might not want to give up a piece of their heart for this analysis. And so that's a really big challenge that we're kind of facing, is we don't really have a way to answer those questions until we can build non-tissue-based models for the disease, and this disease and other diseases, I suppose, in general.
Yeah, absolutely. Thank you. Next question, another question from Danuta. She asks, one more, if I may. Do you see MLC2V deamidation at asparagine N converting to aspartic acid at position 14, which next to the phosphorylation site at serine 15?
That's a great, very specific question that I actually have not looked into. So for deamidation, for the specific instrument that we've used, we might not be able to see it. It might not have quite the resolving power for a deamidation, but I actually haven't looked. I can certainly go look and see if we can see, you know, a 1 Dalton difference and see if that's there. So I'm happy to talk with you further about that if you want me to follow up about that.
Thank you. Well, Danuta was lying because I then asked another question regarding one-dimensional liquid chromatography separation. Why cardiac acting elutes after MLC2V and before troponin C and not according to its molecular weight?
Yeah, that's a really good question. So so these are all denatured proteins. So they're going to separate based on their hydrophobicity as denatured elongated proteins. So the fact that actin would elute later in the chromatogram using this technique would indicate that it is higher in hydrophobicity in its denatured form than any of the other proteins earlier than that. And sometimes that has a lot to do with its size. It can be correlated for sure, because a bigger protein might have a larger amount of hydrophobic residues. So that's sometimes correlated. Sometimes it's not, though. So it's really just dependent on the makeup of the amino acid backbone because they're denatured. So.
Okay, thank you. Great. The next question from Beta, I think we may have covered already. Can you comment on medication of the patients and example beta blockers? So I think we've covered that question already.
Yeah, so actually the list is it's quite extensive for all across just 16 of these patients. We have a ton for the treatments for all these different patients, and we're going to publish all of that information once we have this paper out into the world. There are definitely like beta blockers. I couldn't even begin to probably name all of the different types.
Thank you. So it's just a couple of comments from Sarif Guna and Lucy Carey saying thank you, very nice talk. Thank you for listening. And a final question from Satish Kumar just asks if are the donors male or female, or are they a combination?
It's a combination. So we have a combination of male and female for both donor and HCM, and then also there's different ages in the groups too.
Did you do any sex-independent analysis to see if there was differences between males and females, or was there not enough samples really to do that?
We didn't do that. I think it's definitely important to do. And I'm not sure if there would be enough samples. There's 16 and 16. So I'm not sure if we would be able to do that analysis or not with that small of a cohort, but there are definitely plans we're trying right now as we speak to get more and more of these HCM samples so we can maybe do like a cohort of 50 and really start to see, but it just of course takes time to collect these samples, and that's something I think that is very important to look at.
But Trisha, are there differences between male and females, full stop? You know, like in cardiac electrophysiology, there are clear differences between male and females. Are there differences in proteome?
Yeah, that's a really good question. It's something I haven't looked into, and I think I should, and see, you know, are we able to group the phosphorylations even though they're so close in the cohorts? Are there like clear differences between those two groups? That's a really awesome question. I think we should definitely look into that. Something we can easily do.
Well, I think.
Yeah.
Okay, excellent. Thank you very much, Trisha. You've answered at least 29 questions.
Wow.
And so, yes, really great discussion, a great talk. Thank you very much.
Thank you, guys.
And very good luck with your PhD defense.
Thank you.
Yeah,
Thank you very much.
I think, Trisha, if you survive this, I think you'll survive your PhD defense easily.
I was just thinking that. I was like, this is a test run for my defense.
This is like a public Viva anyway, so
right. Thank you so much, and thanks for organizing. This was an awesome experience. So
it's a pleasure.
Thank you.
Pleasure. So Trisha, again, I'd like to thank you. Wonderful talk, impressive responses to questions. I can't believe you still haven't got your PhD. Anybody who takes you on in their lab is going to be very lucky, I feel.
Thank you.
And Emma, thank you once again for your insights and for willingness to share as well. And of course, thank you everyone, and we'll see you hopefully all tomorrow.
Thank you.
Thank you.
Bye.
Bye everyone.